How are intracellular and cell membrane hormone receptors alike, and how are they different?
Hormones can only affect cells that have receptors specific to them. These receptors are located either inside the cell or on the cell membrane, and their location determines how the hormone signal is processed. Intracellular hormone receptors are found inside the cell, either in the cytosol or directly within the nucleus. Only hormones capable of crossing the cell membrane can bind to these receptors. Steroid hormones, which are lipid-soluble, can diffuse directly through the membrane. Thyroid hormones also bind to intracellular receptors, entering the cell by a carrier-mediated mechanism that requires energy and sodium. When steroid hormones bind their receptors, a hormone receptor complex forms and moves to the DNA in the nucleus. Thyroid hormones bind directly to receptors that are already attached to DNA. In both cases, the hormone-receptor interaction activates transcription of specific genes. The resulting mRNA exits the nucleus and directs protein synthesis. This type of signaling changes cellular activity by altering protein production. In contrast, cell membrane hormone receptors are used by hormones that are hydrophilic and cannot cross the lipid bilayer. Almost all amino acid-derived hormones, except thyroid hormones, use these receptors. When these hormones bind to receptors on the cell's surface, they do not directly alter gene transcription. Instead, they activate intracellular signaling pathways through second messengers. The most common second messenger is cyclic AMP. AMP stands for adenosine monophosphate. Hormone binding activates a G protein, which then activates adenylyl cyclase, an enzyme that converts ATP into cyclic AMP. A G protein, short for guanine nucleotide binding protein, is a molecular switch inside the cell that helps transmit signals from a hormone or neurotransmitter at the cell surface into the cell. So after adenylyl cyclase converts ATP into cyclic AMP then activates protein kinases, which trigger a phosphorylation cascade and alters cellular activity. Another major pathway uses IP3 and DAG. IP3 is short for inositol 1,4,5-triphosphate, and DAG is short for diacylglycerol. In this system, phospholipase C splits a membrane phospholipid into DAG and IP3. DAG activates protein kinases, while IP3 causes calcium release inside the cell. Calcium acts as a second messenger, directly influencing enzymes or activating proteins such as calmodulin. The key distinction between intracellular receptors and those on the cell membrane is that intracellular receptors directly regulate gene transcription, while cell membrane receptors use second messenger systems to generate rapid, amplified responses without the hormone entering the cell.